Hello friends, so in this section we are going to discuss neurotransmitter. So neurotransmitter, it's an important topic from sensory physiology. So what is a neurotransmitter? As you all must be knowing, neurotransmitter is a chemical substance which is released by the nerve ending, and this substance it carries the message between two neurons. So message from neuron 1 is carried to neuron 2 with the help of this chemical called as neurotransmitter, or this neurotransmitter also carries message between a nerve and a muscle in case of neuromuscular junction. Now let us try to understand the classification of neurotransmitters. So neurotransmitters can be broadly classified into two types. These are either small molecule rapidly acting neurotransmitters. So one class is small molecule rapidly acting neurotransmitters, and another class is large molecule slow acting neurotransmitters. So neurotransmitters broadly can be of two types. They can be the small molecule neurotransmitters which are rapidly acting, and large molecule neurotransmitters which are slowly acting. Now small molecule rapidly acting neurotransmitters, this class contains majority of neurotransmitters that we come across. So these neurotransmitters can be divided into four classes. So we have class 1 neurotransmitters, class 2 neurotransmitters, class 3 neurotransmitters, and class 4 neurotransmitters. So there are four classes of neurotransmitters which are small molecule neurotransmitters. Class 1 neurotransmitter, it contains only one neurotransmitter which is called as acetylcholine. So acetylcholine is the most commonly encountered neurotransmitter that we see, and it's a class 1 neurotransmitter. Class 2 neurotransmitter, this class 2 contains all the amines. So norepinephrine, epinephrine, dopamine, histamine, and serotonin, that means 5-HT. We know it's 5-hydroxytryptamine. So all these are amines and they act as neurotransmitters. Class 3, it contains amino acids. So these are also neurotransmitters. So these amino acids include glutamate, aspartate, glycine, and GABA. Now the reason why I'm using a different colored pen here is because the first two neurotransmitters, that is glutamate and aspartate, they are excitatory neurotransmitters. On the other hand, the last two, that means glycine and GABA, they are inhibitory neurotransmitters. Class 4 neurotransmitter, there's only one neurotransmitter in this class which is a gas. It's called as nitric oxide. So these are different types of small molecule rapidly acting neurotransmitters, four classes. Now, large molecule slowly acting neurotransmitters, these are basically hormones which are derived from hypothalamus and pituitary. So, first let us discuss hypothalamic hormones. These are also called as neuropeptides. So, it includes hormones such as uh, GnRH. So, GnRH is released from the hypothalamus and it can act both as a hormone and as a neurotransmitter. Similarly, TRH. Now, it also includes pituitary peptides. So, hormones which are released from the pituitary gland, peptide hormones. These are hormones such as thyroid stimulating hormone, adrenocorticotropic hormone. They can also act both as hormones and as neurotransmitters. So, they are examples of large molecule slow acting neurotransmitters. Now let us try to understand the working of a synapse. So before that we should know what a synapse is. Synapse is a connection between two neurons. So if this is a neuron 1, let us say this is first neuron and here is another neuron, that is second neuron B. So in between these two neurons a connection exists and that connection is called as synapse. So this connection here is called as synapse. So synapse is nothing but connection between two neurons. Now the first neuron, as it comes before synapse, so we know that the flow of action potential is from the dendrites to the axon to the nerve ending. So first neurotransmitter is called as presynaptic neurotransmitter, and the second neurotransmitter to which the action potential will flow after crossing of synapse, that's called as postsynaptic neuron. So in this case neuron A is the presynaptic neuron and neuron B is postsynaptic neuron. Now if we if we magnify it a little bit, the synapse part, so this is the neuron A, and this part, the end part of this neuron, is called as a nerve ending, and this is the neuron B, that means postsynaptic neuron, and the part on which neurotransmitter is going to bind, it's called as the postsynaptic membrane. So we have neuron A and neuron B, nerve ending of neuron A and postsynaptic membrane of neuron B, and in between this small space is called as synaptic cleft. Now this neuron A, it contains in the nerve ending, it contains the neurotransmitter, and this neurotransmitter is present in small vesicles which are called as presynaptic vesicles. So once action potential arrives at neuron A, that means at presynaptic neuron, this action potential is going to cause stimulation of voltage gated calcium channels in the presynaptic neuron. So in the presynaptic nerve ending, we have voltage gated calcium channels, and once action potential reaches at that place, it causes opening of these calcium channels, so there will be calcium influx. Now this calcium in the nerve ending is going to help in exocytosis of neurotransmitter. So as we all know, neurotransmitter is present in the presynaptic nerve in these vesicles called as presynaptic vesicles. So we have presynaptic vesicles, and these vesicles contain neurotransmitter. Now if we take a look at this presynaptic vesicle separately here, this presynaptic vesicle contains neurotransmitter, and in the membrane of this vesicle certain proteins are present which are contractile in nature. So we have contractile proteins present in the membrane of presynaptic vesicles, and once calcium enters, it causes contraction of these proteins, because of which these proteins move towards the synapse, because of which these vesicles move towards the synapse, and there will be release of neurotransmitter by exocytosis. So this is how it's going to appear after exocytosis. So one of the vesicles has fused with the presynaptic membrane, and the neurotransmitter has now been released. So this neurotransmitter is released and it goes into synapse, and now it is going to carry the message from the first neuron, neuron A, to neuron B. Now let us see how that happens. On the postsynaptic membrane, that means on neuron B, certain receptors are present on which this neurotransmitter is going to bind. These receptors are specific for neurotransmitter. Now if this neurotransmitter is an excitatory neurotransmitter, for example glutamate or acetylcholine, then in that case there is going to be increase in sodium influx in the postsynaptic neuron, or increase in calcium influx, or decrease in potassium influx, and all these factors, they increase the net positive charge of the second neuron and result in generation of an action potential called as excitatory postsynaptic potential in the postsynaptic neuron. Similarly, if the neurotransmitter is an inhibitory neurotransmitter, then it is going to cause, after binding to receptors, there will be either decrease in potassium efflux or increase in chloride influx. So there will be net gain of negative charge, and thus there will be development of an inhibitory potential on the postsynaptic neuron, which is called as inhibitory postsynaptic potential. So depending on the type of neurotransmitter, there can be either generation of excitatory postsynaptic potential or inhibitory postsynaptic potential. So inhibitory neurotransmitter, it can be glycine or GABA. In case of excitatory postsynaptic potential, the neurotransmitter can be acetylcholine, glutamate, and this excitatory postsynaptic potential, since it increases the potential of the neuron, it can lead to generation of action potential in second neuron. So if, let us take an example, this neurotransmitter is acetylcholine, so it binds to acetylcholine receptors, and these receptors cause sodium influx. So they are sodium channels. Once sodium enters into the second neuron, it causes generation of action potential in the second neuron. Thus the action potential from neuron 1 goes to neuron 2. Inhibitory postsynaptic potential cannot cause generation of action potential. Now let us discuss another important topic from sensory physiology, that's the types of inhibition. There are 5 different types of inhibition. So first type is called as direct inhibition. It's also called as postsynaptic inhibition. So what is direct inhibition or postsynaptic inhibition? So let us try to understand it with the help of a simple diagram. This is neuron 1 and here is neuron 2, and in between these neurons there is formation of a synapse. Now in case of direct inhibition, neuron A, which is the presynaptic neuron, it will directly inhibit neuron B, that means postsynaptic neuron, by causing release of an inhibitory neurotransmitter, and in this case the inhibitory neurotransmitter happens to be glycine. So neuron A is directly going to inhibit neuron B by causing release of glycine, and this causes generation of inhibitory postsynaptic potential in neuron B. And as we all know, inhibitory postsynaptic potential, it increases the potassium efflux and it can cause hyperpolarization. Thus neuron B will be inhibited. So action potential cannot be generated in neuron B or postsynaptic neuron. This type of inhibition is called as direct inhibition or postsynaptic inhibition. We have another type of inhibition which is called as indirect inhibition. It's also called as presynaptic inhibition. So what is indirect inhibition or presynaptic inhibition? Let us try to understand it with the help of another simple diagram. Let us say this is neuron A, it synapses with another neuron B, and this synapse, this is neuron A, this is neuron B. So neuron A, it causes stimulation of neuron B, that means excitatory neurotransmitter will be released here and neuron B will be stimulated. But there is another neuron which is inhibitory in nature, and it forms an exoaxonal synapse with the presynaptic neuron, and this neuron is inhibitory because it releases GABA. So once this presynaptic neuron is inhibited, it will cause less excitation of postsynaptic neuron. So less excitation of neuron B will be seen. So thus we can say relatively neuron B is inhibited. Now some important things that we need to remember about this type of inhibition. First thing is that GABA is the inhibitory neurotransmitter involved. Another thing is that the synapse which is formed is called as exoaxonal synapse, and this synapse is found between inhibitory neuron and presynaptic neuron. Exoaxonal synapse. So this inhibits presynaptic neuron, so there is less excitation of postsynaptic neuron. Now if we take a magnified look at this area, so this is the neuron A or the presynaptic neuron, and here is the inhibitory neuron, and this neuron, it causes release of GABA which acts on the presynaptic neuron, but it forms exoaxonal synapse, and this is the postsynaptic neuron or neuron B. Neuron A itself causes release of excitatory neurotransmitter which excites neuron B. So this is what we discussed, and this inhibitory neuron inhibits neuron A. Now how does this inhibitory neuron inhibit neuron A? As we studied that the neurotransmitter involved is GABA, so on the presynaptic neuron the GABA receptors are present which cause generation of inhibitory postsynaptic potential in the presynaptic neuron. These GABA receptors are of two types. They can be either GABA A receptors or GABA B receptors. GABA A receptors, these are nothing but simply these are chloride channels, so they will increase chloride conduction. That means more chloride is going to enter into this neuron. And GABA B receptors, they increase potassium conductance, that means there will be more potassium efflux, and both these factors will cause hyperpolarization of presynaptic neuron. Thus this neuron will be inhibited and it will cause less stimulation of neuron B. So this is what we studied in a bit more detail. Now let us discuss another type of inhibition, that's called as the feed forward inhibition. So third type of inhibition. So before that, let us discuss the example of this indirect inhibition. So it is seen in the motor pathway. When we discuss motor pathway, you can check out the video of motor pathway. It's present in a separate playlist. So this neuron is called as alpha motor neuron. Alpha motor neuron, it's also called as lower motor neuron, and it goes and supplies the muscles of the body, and as a result there will be contraction of different muscles. Now this upper motor neuron, it causes contraction of muscles when it is stimulated by a neuron from the corticospinal tract. So this lower motor neuron is under the control of corticospinal tract, and this neuron is called as upper motor neuron. So upper motor neuron causes stimulation of lower motor neuron or alpha motor neuron, only then there will be contraction of muscle. Now There is another neuron present in the spinal cord which forms exoaxonal synapse with the upper motor neuron, and this being an inhibitory neuron, GABA releasing inhibitory neuron causes inhibition of this neuron. So this type of inhibition is what we studied just a while ago, that is indirect inhibition or presynaptic inhibition. So it is seen in the descending motor pathway, and the significance is that it helps in controlled release of excitatory neurotransmitter. That means controlled contraction of muscles will be present because of this type of inhibition. So this is the significance of presynaptic inhibition or indirect inhibition. Now let us discuss the third type of inhibition. It's called as feed forward inhibition. Feed forward inhibition. This is a very simple type of inhibition. Let us try to understand it with the help of a diagram. So let us draw some neurons. Let us say this is neuron A, and this neuron synapses with neuron B, and neuron B synapses with neuron C. Now in this type of inhibition, neuron B will cause inhibition of neuron C, and neuron A will cause neuron A will cause stimulation of neuron B, and in turn neuron B will cause inhibition of neuron C. So neuron B causes inhibition of neuron C, and neuron A causes stimulation of neuron B. This type of inhibition is called as feed forward inhibition, and it is seen in cerebellum. In cerebellum, granule cells, they cause excitation of basket cells, and basket cells in turn cause inhibition of Purkinje cells. So this type of inhibition seen in cerebellum is called as feed forward inhibition. Now let us discuss the fourth type of inhibition. It's called as the feedback inhibition. So we discussed feed forward inhibition a while ago. This is feedback inhibition. So what happens in case of feedback inhibition? Let's again take example of the descending motor pathway in spinal cord. So this is upper motor neuron. It causes stimulation of lower motor neuron or alpha motor neuron, which goes to the muscles, and there will be contraction of muscles. Now the lower motor neuron, once it is excited, it gives a collateral, and that collateral causes stimulation of another neuron which is actually inhibitory in nature, and that causes inhibition of the same neuron which stimulated it. So this collateral causes excitation of a neuron, and that neuron inhibits the same neuron whose collateral excited it. So this type of inhibition, this cell is called as the Renshaw cell. This inhibitory cell is Renshaw cell, and this type of inhibition is called as the Renshaw cell inhibition. This is called as Renshaw cell inhibition, and the significance of this type of inhibition is that it helps in fine and well controlled movement of muscles. So there is fine and well controlled movement of muscles because of this feedback inhibition or Renshaw cell inhibition. Now we have another type of inhibition, that's the last type. It's called as the lateral inhibition. Lateral inhibition is most commonly seen in case of retina. So in retina we have photosensitive cells are present, which are called as rods. So a rod cell, let me draw a rod cell for you. So this is a rod cell. So once rod cell receives the light stimulus, so once it receives stimulus in the form of light, this rod cell will be stimulated, and in order to produce better contrast of the image so that the margins of image are visible clearly, this rod cell causes inhibition of the surrounding neighboring rod cells. So this is an example of lateral inhibition, and the significance of this fact is that it helps to improve contrast of the image. So the margins of image are visible clearly because of this lateral inhibition. Another classical example of lateral inhibition is when we discuss 2-point discrimination. So if stimulus is given to 2 points, so the body is able to identify these 2 points as 2 distinct points because their stimulation causes inhibition of the surrounding areas around them. So once the surrounding area that's marked in red color, once it will be inhibited, only then these 2 points will be distinguished as 2 different points. So it's called as 2-point discrimination, and the mechanism is again lateral inhibition. So these were different types of inhibitory mechanisms in central nervous system. Now let us discuss the last topic of central nervous system, that's laws of sensory physiology. Laws of sensory physiology. So there are certain laws which govern the different concepts in sensory physiology. So let us start with a basic law. It's called as Bell-Magendie law. Bell-Magendie law, so this is related to the spinal cord. If we take a cut section of spinal cord, so if this is cut section of spinal cord, these are different horns in the spinal cord. This horn is called as the dorsal horn or the posterior horn of spinal cord, and this horn is called as the ventral horn of spinal cord. So this law states that the dorsal horn of spinal cord is always sensory and the ventral horn is always motor in nature. We already know this because from the ventral horn alpha motor neuron arises, which is motor in nature. Okay. Second law is called as Muller's doctrine of specific nerve energies. Muller's doctrine of specific nerve energies. So let us try to understand this law with the help of a simple diagram. Let us say this is a receptor. So once this receptor will be stimulated, it will stimulate a particular area of the brain or central nervous system, and there will be a fixed pathway for this. So we have a receptor, pathway, and this area of the central nervous system or area of the brain. So Muller's doctrine of specific nerve energy, it states that the receptor, pathway, and the area of the brain stimulated, they are always same, they are specific for a particular sensation. So receptor, pathway, and area of brain, they are specific for each sensation. Okay. Now if we give a stimulus in the middle of the pathway, now this stimulus is going to propagate, the action potential is going to propagate, and the same area of brain will be stimulated, so same sensation will be perceived. Even if we stimulate pathway in the middle, same sensation is going to be perceived. This is the Muller's doctrine of specific nerve energies. Now let us discuss the third law of sensory physiology. It's called as law of projection. So let us try to understand this law, that's law of projection, with the help of same diagram. Let me draw the same diagram for you. This is a receptor, this is the central nervous system, this is the area of the brain which is involved, and this is the pathway. So for this sensation, law of projection states that if the pathway is stimulated in the middle, the sensation, obviously the sensation is going to be same as per Muller's doctrine of specific nerve energies, but this sensation, it appears to be arising from the site of the receptor. This appears to be arising from the site of the receptor. So we did not actually stimulate the receptor, we stimulated the pathway, but the origin of this sensation appears to be same as the location of receptor. So this is very significant when we try to understand the concept of phantom limb. So what is phantom limb? If we have a fresh amputee, that means the leg of the foot of this person has been amputated. Now if we have a fresh amputee, the nerve will also be cut along with the cut part of the leg. So the nerve is also going to be cut. So here is the cut end of the nerve, this is cut end of the nerve, and because of inflammation there is going to be chronic stimulation of this nerve here, and that is going to cause generation of action potential. So the pathway has been stimulated somewhere in the middle, and this causes stimulation of same part of the central nervous system, and same sensation, that is pain, will be perceived. But this pain, it appears to arise from the original site of the receptor. That means it appears to be arising from the foot, but this foot is non-existent because it has been amputated. So pain appears to be arising from non-existent foot. In case of a fresh amputee, this is called as phantom limb, and it can be very easily, very clearly explained on the basis of law of projection. Now the fourth law of sensory physiology, it's called as Weber-Fechner law. So Weber-Fechner law, it's more like a mathematical equation. So as per this law, the response is directly proportional to log of strength of stimulus. That means R is equal to K into log S. K is the constant of proportionality. So it means that if we want that response should become 2 times, for that the stimulus has to be increased 100 times. So that log of 100 is equal to 2, so response is automatically going to become 2 times when S is made 100 times. Similarly, if we want that if the strength of stimulus is increased 1000 times, the response is going to become 3 times of the original response. Now let us discuss the last law of sensory physiology. It's called as Stevens power law. So as per this law, again the response that is produced is equal to k into I raised power a. I is the intensity of the stimulus and k and a here are constant. So this is Stevens power law. R is equal to ki power a. So that was all about today's lecture. I hope you liked this video, and thank you.